New study maps out the timeline of DNA damage for multiple myeloma. Newswise, Miami, Florida, August 20th, 2025. A new study, https://www.nature.com, articles/4158-8-02921. Maps out the timeline of DNA damage for multiple myeloma, the second most common blood cancer. The findings may lead to better ways to group patients by the state of their DNA and define new subtypes of disease to better predict treatment strategies and outcomes. Better definition of biological subtypes of multiple myeloma is critical for the development of precision medicine treatment strategies, said study author C. Ola Landgren, MD, PhD, director of the Sylvester Comprehensive Cancer Center at the University of Miami. The goal is to optimize clinical outcomes for patients. The findings also support previous estimates of a very long time frame for multiple myeloma development. The first initiating genomic events can occur as early as 4 decades before diagnosis, the new data reveal. The study was conducted by multiple institutions, including Sylvester, Memorial Sloan Kettering Cancer Center, MSKCC, and the German Cancer Research Center, DKFZ. Each institution has its own unique skill set, and our institute is well versed in computational biology. Co-first author Marcela Cadora emphasized the importance of analyzing a large data set involving 382 multiple myeloma patients. She focused on extracting key timeline information from 421 whole genome sequencing profiles obtained primarily from newly diagnosed patients. These profiles provide snapshots of mature disease and were obtained from both newly diagnosed and treated individuals. The trick was identifying historical data within the genome. Genes contain a variety of DNA alterations. Which ones came first and which next? To answer this, Cadora and colleagues used a method called the molecular time model, developed partly by Francesco Mara, an MSKCC physician scientist. This model leverages an internal molecular clock in the genome. DNA accumulates a point mutation, such as a C turning into a G, in a steady rate. Each point mutation represents a single change in the AGCT code of DNA. While some mutations may be harmful, nearly all are benign. Benign mutations often arise when a chromosome goes rogue and duplicates itself, leading to tumor development. After this duplication, the extra chromosome diverges and accumulates unique benign mutations over time. The number of these mutations indicates the age of the extra chromosome. A low number suggests a young chromosome, while a high number indicates the chromosome has been around the cell for years, accumulating more benign mutations over time. The model incorporates this information to estimate the timing of specific DNA damage events. Translocations, or fragments of DNA attached to new chromosomes, are calculated similarly. The strength of this kind of analysis is that it puts cancer-driving mutations into a clinical and temporal context, said study author Benjamin Diamond. In other words, we can effectively put an absolute timestamp estimate on when an aberration occurred, he added. Multiple myeloma typically develops from an asymptomatic stage, MGUS, through smoldering multiple myeloma, and finally to full-blown disease. This progression mirrors the accumulation of DNA damage. The researchers identified several key genomic events that often occur during this process. These include hyperdiploidy, 2 extra chromosomes, canonical IGH translocation, transferring a gene region to a cancer-promoting site, and the duplication of the long arm of chromosome 1, CHR1Q gain. Major findings include IGH translocation was the key initiating event in patients with hyperdiploidy. IGH translocations always preceded hyperdiploidy in these 10% of patients. Patients who acquired Chrome1Q gain early in disease fared worse clinically than those who later did. This finding suggests Chrome1Q gain could serve as a prognostic indicator for patient outcome. Chrome1Q gain also occurred in response to melphalan, a drug used prior to stem cell or bone marrow transplantation. DNA damage initiates many years before disease becomes evident. Consistent with other studies, initiating events typically occurred when people were in their 20s and 30s, decades before diagnosis, their 50s and beyond. Until recently, I don't think anyone really appreciated how early these events are occurring in some patients, said Marcela Cadora. The new study raises questions for future research. Can the DNA damage timeline reveal additional potential prognostic indicators beyond chrome 1q gain? How might early DNA damage events influence subsequent events? How do resistance mutations arise after treatment? In the future, researchers may develop a version of the molecular time model suitable for the clinic. Perhaps the model could be used to estimate patient survival or even potentially guide treatment. There is power in the timing of when these events occur in multiple myeloma, said Cadora. It's not just about what the tumor is, but how it became that way and when. Read more about Sylvester research on the Inventum blog. https://newsmedmiami.edu